Hello friends, in this lesson we continue to human genetics. So we're going to study on about human chromosomes, how to reduce the effect of some genetical diseases and some information about the genetic counseling. Human chromosomes, which are not directly related by determination of the sex in human, are called the autosomes, body chromosomes, and they are 22 pairs, like 44 plus X for female, and 44, which means 22 pairs of what? Autosome. Remaining pair, as we know, represent the sex chromosome, which are X and Y in male. Chromosomes of human many different in shape according to locale or position of centromere. We know centromere is the point which connect two sister chromatids to one another. In a metacentric chromosome, so we know from metaphase, meta means center, so the position of centromere is center of chromosome. Centromere is located at the center, as we see in this picture. Locus of chromosome at the same size. In B, submetacentric chromosome, centromere is closer to one of the poles and locus is in different size. So the position of centromere is not in center, but closer a bit above or below the center of chromosome, submetacentric. And C, acrocentric chromosome, centrosome more closer to one of the poles, and look at as Y during the cell division in acrocentric. We see the centromere relatively closer to the top of chromosome. Telocentric chromosome, centromere located totally at one of the poles. Yes, so 4 different types of chromosomes according to position of centromere. On the basis of the length of the chromosome and the centromere's location, the chromosome of human can be ordered in 7 groups of autosomes from A, B, C, D, E, F, G, and 1 pair of sexual chromosome, XX or XY. On that base, so 23 pairs of chromosome in body cells divided into following groups, like chromosomes number 1, 2, and 3, group A, 4 and 5, group B, 6, 7, 8, 9, 10, 11, 12, group C, 13, 14, and 15, D, 16, 17, 18, group E, 19, 20, group F, number 21 and 22, group G, and sex chromosomes, group X, which determine the sex of organism. And we can observe from this picture also how the shape is look like or different one from another. Diagnosing the genetical diseases. People who have got family history in getting affected by a genetic disease usually have the genetic examination, especially before having children. They need to do these tests or examinations to prevent suffering of next generation from this kind of diseases. But there are too much to diagnose these diseases. The method of amniocentesis, so they take sample from amniotic fluid, the fluid which embryos swim in mother uterus, because it contains the cells as part of embryo, so genetical study of cells can diagnose the disease. In B, method of the chorionic villi. Chorionic villi, as we know, the uterus, then placenta, and the umbilical cord, and then embryo. So in this order, the place which placenta connected to the mother uterus, because mother uterus inner wall is not smooth but folded, and the placenta provide nutrients or exchange material between embryo and mother, so the small blood vessels connected make what like cytoplasmic extension to connect to the mother uterus. So this villi, like finger-like projection, so they call it chorionic villi, which connect embryo to the mother uterus. So take example of them is a method for diagnose the genetical diseases before birth or before symptoms appear. Reducing the symptoms of some genetic diseases. That can be done in many ways. You remember some of them we have been studied in chapter 4, especially about the embryo. So some drugs can be given to the mother to correct or decrease the effect of disease. Also, they can do some surgery, which can be done only in some special hospitals because it's too sensitive. So how to reduce some symptoms of some genetical disease? By diet. This is described for genetic metabolism disease such as phenylketonuria. So about what? About protein. So we can control our food ingestion, taking less amount of protein, so less amount of this chemical substance in blood, and decrease the effect of disease. Physiotherapy, this is described by cystic fibrosis patients, a disease about respiratory organs, lungs. So the patients have many sessions which hitting on the back and the chest will be used to get a sticky mucus from the lungs. And number 3, using certain injection for some disease such as insulin injection, which are used to treat the urine diabetes, about disease which there is no enough insulin hormone produced, so it can be decreased by what? Taking insulin injection into the blood, or what? And the injection of blood clotting protein to treat the hemophilia. Using some surgical operation for the embryo in limited cases to repair some genetical abnormalities, like sometimes there is a hole between two left and right side of heart, so it can be corrected by surgery, which is a symptom or effect of some genetical diseases. And 5, treatment by the genes, exchange the gene which suffer of problems in function of mother and father. Do the test, so the genes which cause diseases are determined and they can be changed with some other genes. That's by reducing the symptom of disease which original gene is responsible for it. Like the removing the gene which causes the diabetes disease, replace it with a normal one. Genetic counseling. So anything the scientists do for what? For benefit of humanity. They use them to help people. How genetic can be used. Many people with a family history for genetic disease also undergo genetic counseling, the process of informing a person or couple about their genetic makeup. Genetic counseling is a form of medical guidance that informs individuals about the problem that might affect their offsprings. By studying the data from the genetic screening test and the family pedigree, the genetic counselor can predict the likelihood that a couple will produce an affected child. For diseases that have both genetic and environmental influences, diabetes, physicians and counselors can advise families on how to lower the risk factors, because prevention is more important than repairing. The fields which can be cancelled know the range of affecting some members of family by one of the genetic disease, so they give information to the couples about the genetic problems made their offspring can suffer in the future. 2. Know what leads to possible genetic disease, especially in relative marriages. We know about about the recessive genes. So recessive genes is more likelihood to be found in the same person which are closest to one another. Know the causes of irregular sexual information or late sexual maturity. Give advices in case of frequent miscarriages. So in case of identifying the father, so not to switch their baby in hospitals. In case of finding out the danger of medicines and radiation, so they can take the samples from embryo, use them, or stem cells, use them in genetical experiments, or to know the effect of medication and radiation, and then give information to the people about effects or side effects. Human genome. After a half century of discovering the structure of DNA by Watson and Crick, genetic scientists knew the genetic successive or human genome, which includes order of about 3.3 billion pairs of nitrogenous bases in human chromosomes. Scientists, but knowing this nitrogens like knowing the letters in a book, but not their order and not what it's mean. Scientists looking forward to know the information which the nucleotide succession will determine for DNA, real terms. They're still looking forward to what these words mean. That's by developing new and important field of biology, field like bioinformatics, which is aiming to program the computer to help to explain, analyze most of DNA nucleotides, their successive, and expect genes presence places, the function which controlled by it, also comparing between succession of different nucleotides for DNA. So to know the sequence of nitrogenous bases, which genes they make, what the function of gene, what it affected by, and how show their effect on what, on phenotype of organism.